Hi, my name is Lisa Lai. I'll be presenting a talk entitled Hackathons, a new space for collaborative projects and learning. So I think it's really important to give this talk today because, the number of people attending OHBM Brain Hacks and Brain Hacks in general has really been going up. I mean, last year we had 200 people. This year, given the fact that our event is completely virtual, we'll have 500 people, which is amazing. But as you all know, 500 people is still a small fraction of the total OHBM community. So I'd like to use this as a chance to tell you a bit more about what we do, perhaps convince you that a hackathon is worthwhile to attend, and perhaps you'll even be inspired to host a hackathon yourself. So I'll be going over what a hackathon is. I'll be talking about the compatibility between hackathons and academia. I'll then talk about the larger brain hack movement, why and how to participate in a brain hack event, and I'll also be presenting a case studies of a trainee's experience attending multiple brain hack events over the course of their degree. So I think that the best way to convey what a hackathon is to actually just show you what it looks like. Means here you see groups of people sitting at different tables and they're all co-working. This specific photo was taken at an event called Deep Brain Hack, which was held in 2017 in Montreal, Quebec. And at this event, people were all working on projects related to deep learning applications for neuroimaging. And I mentioned that this is part of the larger brain hack movement, but I think that I'll first start by actually telling you a bit more about more traditional hackathons. So hackathons came about in the broader technology sector, and generally they're defined by intensive collaboration with the goal of innovating fairly rapidly. But hackathons come in multiple flavors. Two of the most common ones are competitive community hackathons and brand or corporate hackathons. Competitive community hackathons can be held at high schools, at colleges, and just in broader communities, and they're often centered around a theme. Brand or corporate hackathons are typically sponsored by one or multiple companies, and the ultimate goal is for participants to develop a feature that is ultimately then merged into the product of that company who's sponsoring the event. Something that's common to both of these types is that participants are typically incentivized either by monetary reward or some other type of prize. And additionally, it's important to know that projects are conceptualized at an event very often with traditional hackathons, and they're expected to be completed by the end of an event. So I include asterisks for the last two of these points, because I do want to give a disclaimer. Not all hackathons are like this. In fact, many hackathon organizers have actually been moving away from imposing these unrealistic expectations. Either way, you're probably beginning to think, this sounds pretty different from academia. Are hackathons and academia compatible? So one of the main challenges with organizing a very successful hackathon event is ensuring that there is space for individuals spanning different skill sets and different skill levels. So I believe that for trainees in particular, hackathons are a really great place to, in a safe experimental space, learn about new tools. And the more tech lingo for this is hackathons can essentially just serve as a sandbox for trainees, while also allowing people who are a bit more experienced to come in, to collaborate with others, and build their tools. With regard to the competitive element of hackathons, we all know that academia is not without competition. However, science is ultimately improved by collaboration. And as I'll try to convey in the next few slides, the reward of participating in a brain hack isn't a monetary one or a prize. It's more about bridging gaps between different institutions and disciplines, and about developing and continuing collaborations for the greater good of science. And lastly, we know that academic research projects are long-term. The research life cycle is very long. Publishing a paper takes a very long time from when a project is actually conceptualized until you finally have a very pretty wrapped up paper to present to the broader community. Nonetheless, hackathons are a great way to chip away at very concrete goals, and it's becoming more and more the norm that at brain hack events, participants are given a project template that they have to follow. So those who are proposing a project are really encouraged to think long and hard about specifically what they want to get done, to think about more attainable and easily achievable goals. So this brings me to the brain hack movement and format, one that has been developed to be accessible to brain mapping enthusiasts across the spectrum of different skill sets. And there have been a number of different people actually involved with creating this event format. I can't take any credit at all for this diagram or for the format in general. But essentially, the brain hack event is typically split into three parts. You have the hackathon, and this is where inspiration has been taken from what I've previously been talking about, where people come in, they propose projects, and at the very beginning of an event, participants can decide which projects they want to get involved with. And as I already mentioned, those who are proposing a project are encouraged to think about what kinds of skills would really improve their project. It doesn't necessarily have to be technical. The other part is the educational portion. So we call this the train track. And it's where participants can come. They have an instructor who teaches them about different tools. And in some events that I'll actually be talking about later on, participants are encouraged to bring their own tools and have more of a flipped classroom setting where they're encouraged to use these tools that they're learning about in their own work. And lastly, there's the unconference portion. And so this is the more dynamic component of the event, where unlike a conference where people have to submit abstracts and symposia proposals way ahead of time, here participants just decide on the spot what they're going to be presenting. And we all know how interdisciplinary OHBM is, and BrainHack is very much a microcosm of that, merging neuroscience, statistical modeling, and computer science. So looking at this diagram, you might wonder if all projects at a BrainHack event are technical, and that isn't at all the case. Just to give you a taste for the kinds of projects that participants have worked on, I've selected 4 from last year's OHBM BrainHack. So we have seen a massive gamut of different types of projects, ranging from initiatives to standardize study consent forms, such that ethics boards more readily approve sharing of brain data, to a community-sourced checklist of everything that must be included in a neuroimaging paper, such that whoever reads that paper can very easily reproduce the results. And there are more technical projects, of course. Last year, Talia Racconi presented a project which would be a JavaScript toolkit for modular brain visualization, as well as an automated cortical lesion tool using machine learning. So a machine learning tool for automatically detecting cortical lesions. But the common feature of all of these projects is that they're all open source and they help advance reproducibility and standardization in neuroimaging. And I keep using the term BrainHack, and I want to point out that there are a lot of other events that share a very similar mandate. Some of these events, such as Neuro Academy, which takes place at the University of Washington, focuses a lot more on the teaching component, whereas another event that happened very recently, and virtually actually, BrainWeb, really emphasizes the collaborative hackathon portion of the event. And so this year, our event is happening virtually. I'd like to now preface this slide by saying that I'm not trying to make a claim here that the BrainHack format is superior to traditional conferences. However, I do see it as being complementary to the format that many of us are accustomed to. So conference symposia and poster sessions are an excellent space to present a polished version of one's work. And we know that poster and networking sessions are probably where the most inspiring and insightful discussions happen. BrainHack events provide a venue to actually put into practice and actualize a lot of these ideas. And the educational components of BrainHacks tend to be quite interactive. They're an excellent space, as I mentioned previously, for trainees to come in and learn about new tools while getting help from those who are more experienced, if not even the actual developers of these tools themselves. So with that in mind, we can think of BrainHack events as really accelerating the adoption of open source tools for neuroimaging data processing analyses. But beyond that, BrainHack events really emphasize, they're very social in nature, and they're a great place for learning how to work as part of a team, how to problem solve and interact within diverse groups. So I'd like to now actually give you a more concrete example, a case study of a trainee's growth. So let's take a trainee who's just starting out in their master's. They come from a background in cognitive science from their undergrad. They have some computer science experience, and they don't have any neuroimaging experience when they're just starting their master's. So in the first semester of their degree, they attend a BrainHack, and they really take advantage of all of the different training that is available to them. So they learn tools for virtualization, such as Docker, the NiPipe workflow for creating neuroimaging tools, and other existing tools for reproducible neuroimaging. By their next BrainHack, and this is, let's say, half a year after their first BrainHack, they've been using the tools that they were already exposed to. Specifically, they've used an open source PET neuroimaging processing pipeline for their master's project. They've actually been using version control for the statistical analysis that they've been doing. And by that second BrainHack, they're feeling more confident in their version control skills to contribute back to the neuroimaging tool during that BrainHack. By their last BrainHack, they really want to contribute back to the community. And so they sign up to teach GitHub and Git for collaborative software development during an educational session. And I'd like to mention that this is actually based on my own personal experience. I just completed my master's. I started it 2 years ago, and I was really fortunate that I was able to attend BrainHack School in Montreal during that first semester. I found that it was very instrumental to just shaping my degree and really allowing me to immerse myself in the open source neuroimaging community. But there are multiple ways to get involved with the BrainHack event, and so I'd like to wrap up this talk by quickly talking about this year's OHBM BrainHack and how it's a testament to the many ways people with varying levels of expertise can get involved. So knowing that this event would be taking place virtually, we really wanted to leverage the fact that more people from around the world could attend than ever before. So to run a truly global event, we decided to have the event take place 3 times across 3 different hubs that are split by different time zones around the world. So it's a difficult task, and to really ensure that this event would be successful, we had to enlist the help from so many different people around the world. And this gave us the opportunity to define very concrete roles for people to get involved. And so some of the roles that we came up with are hub hosts, and those are the individuals that are essentially the face of each of the hubs. They moderate the event. Then we have project advisors. So this year, every single project is assigned a more experienced person who's already been to many BrainHack events. And they can use, and project leads for these projects can go to this project advisor as a consultant, essentially. They can seek advice from them. Then we still have train track instructors and teaching assistants who are available virtually, or you can have video chats with one another. And then we also have a buddy system, where for every single newcomer who hasn't previously been to a BrainHack, they're paired with another individual who's a bit more experienced and can be available for chats. So to summarize, I hope that I've convinced a lot of you that collaboration is really at the core of the BrainHack community. And this format allows for individuals from the early trainee level to the more senior level to participate in different capacities and grow. And I believe that BrainHack events have a potentially important role in the scientific community alongside the more traditional conferences. And I hope that I've convinced those of you who are watching this talk who've never been to a BrainHack before to check out the next one that's happening locally or the next one that's happening virtually. And for PIs who've been hesitant about whether or not BrainHack events are a good use of your trainees' time, I hope I've convinced you to let them give it a shot. Thank you. Hi, so I'd like to thank everybody who's been involved in the BrainHack community. It started in about 2012, and there are just so many people who have really contributed to creating the format that we know now. So it's impossible to really thank every single person, but here I've chosen photos of those people who were involved in writing up the paper that's published in GigaScience on the BrainHack format, people who have been involved in creating the train track component, those people that were involved in the last 2 years' events, and everybody who's involved in this year's event. Thank you.
Diversity and inclusion are key parts of the open science movement. The more diverse the population who can access, repeat, build on your research, the more impactful your findings or method will be. When we discuss diversity and inclusion, we must consider how different factors intersect to impact the individual. These factors include culture, race, class, language, along with other factors such as gender, sexuality, age, education, and disability. Each of these factors individually form a structure within which one side is powerful and the other side is powerless. These power structures determine dominance hierarchies, where some people's needs are prioritized and others are not. Our conversations about open science, both here at OHBM and also at our home institutions, also function within the same explicit and implicit dominance hierarchies. For the majority of us at this meeting, open science is led by the powerful global north, primarily Western Europe and the United States, while those in the global south have our priorities, models, and methods thrust upon them and are expected to keep up. This does not mean that we should halt the development of our own open science practice, but rather this is an invitation to improve our awareness of issues around access, power, and humanity, and reflect upon how these issues may impact our attempts at improving global inclusion. In the rest of this presentation, I've invited contributions from colleagues with experience of living and working in Kenya, Nepal, Argentina, South Africa, and Bangalore. I have invited them to share their passions around open science, describe some of the difficulties they face when engaging with the global north, and share their ideas for actions which would help them play an active role in driving the open science agenda in a way which works for them. This isn't a lecture on global inclusivity. This is just a trailer to spark some interest, open some questions, and see if we can collaboratively create ways of doing truly diverse and empowering international open science, which works for everyone.
Open science is the future, and it can be a great way to set us free from all the geographical boundaries that we have now.
For me, open science means even with financial limitation, as a student or as a researcher, that shouldn't be a limitation to you accessing scientific research.
For my community, I like to create awareness on the importance of open science.
I also integrate open science or openness in several projects, whether it's a science project or any other project, to achieve the goal fast and efficiently.
If we're open about the things we learn and the way we do it, we can get more people to jump into the same problems and bring more creative ideas to the same challenges.
I have seen many open science platforms around the globe inviting participants in the research. I feel such programs are lacking proper publicization and outreach to reach more and more people.
The main barrier has been with a visa. That's the major barrier when you want to attend workshops or conferences.
It's a very taxing process, both emotionally and financially. So some of those initiatives you just let them pass by.
Previously, I've been barred from joining an online event simply because maybe I was from Africa, or I don't hold a PhD position, or maybe I'm not a postdoc, which I found very discriminating and not good for the open science, in that open science should be inclusive of everybody in the community.
My level of English wasn't very good, so I usually caught 70% of what people said and try to infer the rest.
The difficult part is, as open science based in Kenya, at times I join these communities that's in North America and Europe. They have like advanced knowledge of what open science is, but I just find yourself like, what were they talking about?
I don't think there is a global open science practice. I think we all appropriate open science differently according to where we work, how we work. It will probably become the default in the near future. I think what's interesting is to start thinking how the default will look like, how transformative it will become.
Most of my engagements in open science in Europe have been through summer schools, conferences, and symposium. So, well, I've actually had an opportunity to see some really excellent work actually going on in terms of open science. What I like about some of those forums is actually that you actually see how researchers and institutions are actually taking open science quite really seriously.
So one thing I consider important in terms of ethics is to always have a situated approach, and demanding the same thing to everyone no matter what is assuming all stakeholders producing scientific knowledge start from the same place.
Societies that share data are actually viewed as leaders. Those that don't are actually viewed with a lot of suspicion.
We cannot just leave the excluded to compete at a level where we, as the global north, have all the cards in our hand, have the power, and have the system that supports keeping us in that power.
I think the first time I read about situated openness was from OCSD.net. And I think that's the way to go if we don't want to reinforce inequality in the name of openness.
We have to understand that to participate in any space, we need a certain level of resources and capacity. So a truly open science project needs to be designed to ensure that the barrier to participation is lower.
Good thing nowadays, most conferences are trying to have both in-person and online attendance, so it makes it easy. Even if I miss going there in person, I can still access what's going on online.
The challenge there is that we should work harder to include these voices. We cannot just say we have organized a meeting, a conference, or a symposium, and it's open. We share the videos, we share the slides. Now it's open science. I want to have a community of global open science practice where people are actively reaching out to others instead of just passively sitting and waiting for diversity to happen. It is not a thing that you wait for, it is a thing that you have to work towards.
So one of the examples that really left a huge impression on me is a few years ago when I was invited to go to Argentina to do training around protein and bioinformatics and basic programming. I knew that they didn't need me to teach this. There are people who actually know these. And for me to be there and assume that my knowledge is unique is really, really egocentric.
If there could be a model of partnerships where some of those trainings actually held here in Kenya or maybe here across Africa, where actually most people would actually benefit.
We need to know that the scientists who perform in countries like in Europe and the US, they have privileges like higher power, wealth, and resources. And therefore, they don't need to assume that they need to help the global south. They don't need to help. Researchers in developing and underdeveloped countries have better understanding of their research, and they have the solution to the problems that they face.
What I've realized is I'm passionate about having localized scientific knowledge. So with open science, it means scientific knowledge that we'll be having is for Kenya by Kenyans.
There's also a challenge in terms of the background of people who have not experienced this way of living or this societal problems in the global south. So you don't necessarily have the context for understanding that if you want to scan someone in a remote place, there's like zero infrastructure for electricity, for getting clean signal out with whatever modality you want to use. You have to first address those challenges before you can even think of the medical challenges or the research question, etc.
When working openly, one thing I've realized you have to understand is the social culture, societal culture of the place. So you must understand how this community operates. So even if you have like global knowledge of what open science is, but still have to understand the communal structures that you're dealing with.
I think we understand science as a human endeavor, but up to now it's been involving a very limited portion of the humans in the world. So I get excited about science when I think of everything we could achieve if it was open.
My vision for the future of the global open science practice is, I say it is the future. However, it is also challenging. We are dealing with people with the different minds, but also with the different culture, colors, trends, races, caste. So it should be taken care very well. Let's collaborate. We have always felt to show our socialness or humanness in some way when talking about country, race, caste, color. But open science is a chance to prove that we are social and we are never broken, but united to solve any problem that exists around.
What I would like to achieve for this community by practicing open science is to get everyone on a similar page, where the page is that we are all humans and it's okay to show your humanity.
My vision is that when an open science advocate or practitioner is talking about open, a person in Kenya should say, yes, that's the voice. That's the voice. You share the same sentiments. The same thing. If someone from Kenya is talking about open, then someone in Asia should say, yes, that's what I've been talking about.
It's really cool for me to see that people from the global south are engaging more, and you feel this kind of feeling of that we are working towards a more socially just society if you see that happening.
Open science should work in global solidarity with people. It should offer equal access to information. It should aim to empower each other so that people can use their knowledge to benefit their community and society. irrespective of where they are.
Three, two. Hi everyone, I'm very happy to be here today with other officers of OHBM Open Science Special Interest Group to talk about open science and how we think it can shape the way we work together as a community. By way of an introduction, I would like to talk a bit more about open science. Not so much about what we mean by open science, but rather why it really matters and why it matters for our science in general. If we are all here today attending the annual meeting of the Organization of the Human Brain Mapping, it's because we share a common goal. The OHBM community is very rich and diverse. We have people coming from very different disciplines, going all the way from psychology to computer science. But we all have one common target. We want to build a better understanding of how the human brain works. And in order to do that, we work with data. These data being brain images or brain signals. And I always like to go back to this chart that I've extracted from a publication by Paul Drack and colleague in Nature Neuroscience in 2017. On this chart, you see sample sizes as a function of time. And I really like it because it gives us a good overview of how our field has been doing in terms of data science. As you can see, the chart starts in the mid-1990s, which is a nice start because it's the beginning of functional MRI, a technique that is widely used in our community, up to a few years back, 2015. And the first thing we see when we look at this graph is that, well, yeah, sample sizes have been constantly increasing, and this is very good news. But more importantly, if we look at the last point on this curve that corresponds to 2015, well, we see that in 2015, the median sample sizes across studies was 30 participants. And if we go back to our target, we are studying an organ that is very complex, the brain. We look at a population that is extremely diverse and variable, the human population. And because our field has been going on for quite some time right now, the question we want to ask are likely to display subtle effects. So what do I mean by this? Let's say I want to better understand a pathology. For instance, in the context of Alzheimer's disease, I might want to study changes in the brain that happens very, very early on in the pathology. Early on, because that's a period where it might be easier to find ways to interfere with how the pathology is going to progress. But because it's early on, it's very likely that the changes that I want to look for in the brain are very little. And with 30 participants, it's just very difficult or completely impossible to answer questions that look at very subtle effects. And that's why researchers have started a few years back, let's say 10, 15 years back, to look into open data. And this is not just, of course, not just for neuroimaging, it's a trend we see in many scientific fields that are data intensive. In neuroimaging today, open data takes many forms, including the data that you can share at the end of a study. So including researchers teaming together to build consortium and create larger data sets that they can all work on. A very good example, early example of that was the 1000 functional connectome project looking at resting state fMRI. More recently, we've even seen funders investing into creating shared resources of very high quality that are then made available to the community as a whole. A good example here is the UK Biobank. So all of those open data, part of open science, are very important because they shape the type of question we can ask in our research. And I think open data is one part of open science that is now really well known in our field. Another aspect that I think is important when we think about our studies is to understand what matters and what does not. So what do I mean by this? Of course, it's referring back to the notion of generalizability. When I do a study in brain science, I make many choices along the way. In my research, I'm interested in pipelines. So I can tell you that we make choices about the neuroimaging analysis software that we use. We make choices about the algorithm that we use at a given step, let's say registration. We make choices about the parameters for all those algorithms. But beyond the pipeline, we make choices when we design the experiment. We make choices when we decide on inclusion and exclusion criteria for our participants. And all of these choices will have an impact on our results. On this slide, you can see a figure that I extracted from a paper that was a collaboration with Alex Bowring and Tom Nichols from the University of Oxford, in which are displayed results that use the same data, the same pipeline, but a different neuroimaging software. And you can see that there is quite some variability. Another recent example of that is the NARPS project that you have probably already heard or will heard a lot during this OHBM 2020. In this project, 200 researchers teamed up to analyze the same data set. So they grouped in about 70 teams, and they looked at how the results would be different. And well, you might say, but okay, we are changing the way we analyze the data. We are changing the way we design the experiment. Of course, we expect to get results that are different. Yes, this is entirely true. But I think what's really important here is to which extent those changes are going to impact our conclusion. Will those changes impact the way we interpret the data? And right now, it's really hard to know what is the impact of our methods. It's really hard to know because the way we've been working is that when we publish one paper, we do one study with one method. And authors have suggested that maybe what we could do instead is analyze the data using family of methods, many different methods, get intermediate results, and then pull the final results as maybe a consensus across all those different methods. This is one solution that I personally think is very interesting, though a bit data intense. But there is another approach to this. The other approach is to do what researchers usually do. Basically, we do an experiment to answer a research question. We write up what we think we have found with our study, and then we share that with the community, and other researchers can build onto our work. Here, similarly, we could imagine that if another researcher would think that one of the choices I made can have a strong impact on the results and change our conclusions, they can go and design an experiment that would test exactly that. And this would work quite well, I think. But it's based on the assumption that we have enough details about what happened in the methods of the original paper. And the way we share methods right now in research is through the research paper. So research papers are they provide a great high-level overview of what happened to the data, but they lack the level of detail that we need in order to be able to change just one of these steps and see what happens in the results. And this is why we've seen in the field and in other fields as well, more and more interest into sharing, opening up methods. And this is usually done in the way of sharing codes because the analysis code, it contains a lot of the information we need. So in the first part, I've discussed open science and why open science matters for science. And I've talked about this great initiative. I've talked about 1,000 functional connectome, about UK Biobank, about NARPS. But how can we, as individual researchers, take part in this effort of open science? And before going any further, I wanted to start with my takeaway message. Every little bit counts. So open science is not a 0 or 1 approach. I'm going in the following slide to share a few examples of what you can do to work openly in our field, but there is no need to do everything. Like if in your next paper you do just one step, well, it will enable new possibilities for our community. So just that one step is very important. And maybe the next paper you will do 2 steps, or you will stick with that just 1 step. All is fine. Like open science is a process, and we can go 1 step at a time. So here are a few examples on how we can make our research outputs for a given study available to the community. So here, on the left, I've described a high-level representation of a pipeline. So we start from some raw data, we do some pre-processing, we get derived data of some form, and with the statistical analysis, we finally get our conclusions. This is a very high-level representation. I've used fMRI as an illustration, but this is true of like any brain science study, and even beyond that, of any like data science pipeline, I would say. Okay, so we have this pipeline and we are researchers. So what we like to do is write up a paper that describes our main findings. And the first thing we can do in terms of open science is make sure that this paper is available to the broadest community possible. So this can be through depositing in different archives. So some countries have, or institutions have institutional archives, and there are also preprint archives, postprint archives, and so on. The other aspect we've mentioned is open data. So I can also, at the end of my study, go back to my raw data, make a nice package with it. And BEAT has been a standard that has been developed more and more, and I think is well adopted by the community now, who started with MRI, but now extended to EEG, IEG, and so on. What's nice about BEAT is that it gets, you can create that nice package with not only the data, but also the information that is necessary to enable reuse. So we make this nice bits package, and then we can send it to different databases that are here to make that data available to the world. We've talked about raw data, but basically the results are also data. And similarly, we can package that data and make it available on the database. So here I've used the example of task-based fMRI. We have the IGM standard, and we can upload onto the Neural database and share statistic maps. We can think about doing the same thing for derived data, although here, to the best of my knowledge, there is no single standard that would cover every type of derived data. This is still much in progress, but still you can make a package, share what you can, and make it available on a generic neuroimaging repository such as Nitric. And we've talked about opening methods. So here we would package the code, and we can use databases such as Software Heritage or Zenodo that are particularly well suited to share code. So in the first part, we discuss how open science is important for science in general. In our second part, we've looked a little bit into how practically we can take part in that effort with my research study. But we haven't discussed the most important point, which is really the topic of this symposium. Open science, for me, the most important part of it is that it's a possibility for us to work as a community, to do our research as a community. And I'm really happy to introduce our two next speakers. So the first talk will be from Lisa Levitis, who was the Open Science SIG Hackathon co-chair with Remy Gould this year. She will discuss hackathons and how those unstructured conferences create new opportunities for collaboration and collaborative learning. And then we will hear from Cassandra Gould van Praag, who was the OSSIG Open Science Room co-chair with Stefan Yunis this year. And she will discuss inclusivity, because it's very important that we make open participation possible for the broadest community possible. I had the chance along my open science journey to meet many, many people. And this is just some of them, not all of them are here. I think in particular of communities such as BrainHack, of course, the Open Science SIG within OHBM, the INCF, and Mozilla Open Leaders. And to finish, and this has really shaped the way I do research, but also my research interests. And to end this talk, I would like to thank you, to send a big thank you to all the volunteers who helped with the SIG this year to build the hackathon and the Open Science Group. Thank you very much.
So welcome everyone to the panel discussion of the Open Science in Action Doing Research as Community Symposium. I'm Eugene Duff from Oxford University and Treasurer of the Open Science Special Interest Group. I'd like to welcome also on this panel Camille Maumet of the University of Rennes, who is the Chair of the Open Science Special Interest Group, Lisa Levitis of McGill and Co-Chair of the Open Science Hackathon, BrainHack, which occurs prior to this conference, and Gould van Praag, also of the University of Oxford, and co-chair of the Open Science Room of this meeting. By now, I hope most of the audience will have heard the symposium talks from each of these panelists. The broad focus of this panel is was intended to be on the challenges of open science. It seems a particularly apposite topic given the context with COVID-19, and which has been a real focus of our group in the last few months. So we'd like to focus a bit on this in the panel. I think this crisis has really given us a bit of a fast forward vision to the future of increasing remote collaboration, interaction, increasing use of public and shared data sets and public tools, and certainly more video conferencing than anyone would like to take in any given week. It seems like this moment we're in might give us a unique perspective onto the realities of and opportunities of open science and open neuroimaging, where some of these activities, which were really have been treated as something like experiments at the periphery of our scientific activities, suddenly have gone into the mainstream. It'd be nice, I think, to start with a bit of a discussion of our panelists' experiences about bringing together the elements of our activities in the Open Science Special Interest Group in the context of COVID. I might start with Lisa, who moving the BrainHack hackathon online in just a few months has been a momentous task. But also I think we're aware that we were presented with great opportunities with the online format, greater accessibility, unlimited numbers, and flexibility in our scheduling. How have you found this experience, and has it changed your view on the role of in-person and online hackathons and collaborative events in neuroimaging in the future?
So while I definitely love the opportunity to actually meet up with people in person, there's something that, an online event can't fully replace that. I found that honestly, organizing and participating in an online event is still a terrific experience. There are definitely a lot of difficulties, but what I found is that, especially in the brain mapping community, there's just such a breadth, and there's so many people who have been so enthusiastic to help us out with creating this event. And right, so by now all of you would have seen the symposium, and so we decided to go with a very decentralized virtual event this year. And so we have had 3 hubs. And within these 3 hubs, there have just been so many different people who have been very willing to help out, and they've helped out in a number of different capacities. And so in addition to the OHBM Brain Hack, I've also participated in a different event this year called BrainWeb, which really emphasizes the hacking part and the collaborative portion of the Brain Hack movement. And so with that, I participated in a project where I collaborated with people based in Montreal, as well as in Spain and in Germany. And I found that, yes, of course, there is the issue of time zones, but what I found to be extremely important for ensuring that such a collaborative project is successful when people are in different time zones is one really good and regular documentation of what people are up to, the progress that they're making on their project, as well as a very clear division of the tasks. If you're sitting side by side with different people, you know exactly what the other person is working on. When you are separated across, continents, it's good to know exactly what other people are working on, making sure that there isn't duplicate and redundant effort.
Right, that's great, thanks. Cass, I think you had an equally challenging task in moving the Open Science Room to a remote format. This room is traditionally a very flexible, interactive space at the conference, which is really difficult to replicate online. You also have a role as an Open Science Engagement Officer in your department. Have these few months given you a new perspective on the particular challenges and needs for these types of spaces and the different challenges to maintain them in an online situation?
Yeah, it's interesting that the note about sort of flexibility, like the unconference of the Open Science Room, the sort of organized chaos which everyone loves to be a part of, and it's so important. It creates a real energy and vibe around the place. But when you're managing participation across 3 different time zones, we can't afford to be flexible, really. So we've kind of built in some flexibility, which feels a little bit, not really disingenuous, but we've said, here's a time when you can be flexible. Here's some free play time, go wild. But we still have to be careful about how we ensure that the people who participate in that time are brought in with inclusivity in mind. We don't want these free spaces or any space in the OSR to be monopolized by any one voice. And at an in-person meeting, that's a bit easier to manage because we're all there in the room and you can all sort of get a feel for it. But again, operating across different time zones and things, there are just added complications. But thinking about the way that we're going to be working moving forward at our universities, so our wider imaging group is spaced across 3 different departments and 3 different parts of Oxford. And there's always a bit of a clash about, are we going to hold the meeting in this hospital today or in the other hospital? And then what about the other side that no one ever visits, really? Well, sometimes we go. But they have good coffee over there, so we definitely go. I think we are all now much more used to the idea of working online and remote participation, and used to the idea that these aren't just faces on screens, these are real people, and we're having to work hard to build relationships in this way. But I think if we can bring some of that model into our working when we're back in offices, whenever that may be, it's going to be really helpful for a lot of people. It's about this idea of inclusive design is actually beneficial for many people, not only the people that really need it. We can all benefit from it if we put a little bit more effort into thinking about access needs of different types of people.
Yes, it certainly, it became very starkly aware how it seemed to increase engagement of wider numbers of people with talks and whatnot. But
yeah,
spontaneity is something we had to work to maintain. Yeah. I'm not sure, Camille, if you had any thoughts on your experience as chair of OSIG. I remember we were at the start of planning for the year, we were motivated by becoming a bit more focus towards remotely accessible open science due to carbon footprints, which seems a distant memory now. How did you find your experience?
Well, so yes, we had in mind that we wanted to put more content online, and well, basically here we are. We have to put everything online now, and well, I think I really love the opportunity that this creates to reach out to wider audience. I think this is something we wouldn't have achieved would we have been just doing an in-person meeting. But it's not, I think this like working on these events made me realize that it's not as simple as it would seem. Like we say the event is online and then, well, yeah, it's easier to reach out to different people, but Cass has touched upon the idea of like inclusivity and how we have to put in basically more work if we want to make sure that this is truly accessible to the widest community possible. And I think Lisa was touching on that as well. Like if we want people to be able to collaborate in a hackathon, we need that documentation that will let someone who comes on the next day and didn't have the opportunity to talk with the other member to know where the project is at. So this has like created new challenges for us because we had to redesign everything. And I think it's also related to the fact that, well, virtual conferences are very new for our field at least. And while conferences have been going on for so much time, so people had time to think about it and know, well, it's good to have a coffee so that people can network. And I truly believe that online we can create events that are likely engaging, but it takes effort and it will take time for us as a community to realize how we can actually do all these kind of things. So there are many things we can try, and we are going to, because the hackathon is coming at the time of this recording, try a few of them, but I think more is coming in the future.
Yes, I agree. Just quickly, I might just move back to Cass, speaking about your talk, which beautifully illustrated the potential in the broader world for contributing to science and neuroimaging from places other than the global north. Do you have a more specific vision for how the neuroimaging community might look if we better tap these resources through the sort of addressing the challenges we've been facing this year?
It's interesting. So we've got a couple of talks coming up in the OSR. It'll be in the past, present, and future theme, where we're thinking about the future of open neuroimaging. And two of the speakers I'm really excited about are part of a community that are working on open source hardware solutions. So, I mean, the idea of building a magnet, right, or any type of signal recording equipment for neuroimaging, that's not even entered our arena of thought, because lucky us, we've got a 7 Tesla and a 3 Tesla, and you know, so there are really significant and important challenges that can only be addressed in terms of participant health or patient health, or if you have a magnet. And in countries where you can't afford a 3 Tesla, a 7 Tesla, or 1.5, there are really important efforts going into helping build open source solutions. And there's desktop magnets, and so this is relatively new for me. So in the last year or so, I've been introduced to a few more people in like the world of ISMRM and more physicists and things like that, and they've had this kind of going on for a while. But for me, it's a completely new train of thought that we could take a step back from our position of data collection and analysis and sharing that, but also going back to sharing the actual tools that we need to even start the processes. So I'm really excited to hear about one of our speakers is going to be giving an overview of this whole open source imaging community, and another one is our keynote for that theme is going to be talking about his work in Uganda developing a low field magnet. And then of course that opens up the issues of or opportunities around low field imaging. You know, we're obsessed with going higher and higher, but with high field strength there's new challenges. So there are some situations where low field strength might be beneficial. So it's a specific problem that is being addressed because of a need around money, but the efforts in that direction are going to open up new possibilities and interest for us. So I think for me that's going to be a really cool new direction to take it in.
Thank you. That's really exciting. I think we'll finish this panel now to allow a bit of time to take some audience questions following this. So I'd just like to thank everyone again. It's been a fun few months working with you all, and looking forward to getting to the end of the conference and seeing these open science opportunities really express themselves. Okay, thanks a lot.